or of an administered dose based on a mass balance determination or in comparison to an intravenous reference dose, and an IR drug product is considered rapidly dissolving when 85% or greater of the labeled amount of the drug substance dissolves within 30 minutes using basket method, that means USP apparatus at 100 rpm, or paddle method USP apparatus 2 at 50 rpm in a volume of 900 ml or less in each of the following media. First, 0.1 normal SCl or simulated gastric fluid without enzymes. Second, a pH of 4.5 buffers. And third, a pH 6.8 buffers or simulated intestinal fluid without enzymes. The biopharmaceutical classification systems provides a new perspective to the dissolution testing. It provides scientific rationale to lower regulatory burden and justifies a biowaiver under certain circumstances. The BCS biopharmaceutical classification system also predicts the possibility of obtaining an in vitro or in vivo correlation. Justification of a biowaiver is based on a combination of the BCS classification of the drug substance and a drug product dissolution. Profile comparison. Prerequisites for dissolution based biowaivers are specially to obtain a biowaiver for an immediate release generic product. The reference product should belong to class 1, high solubility and high permeability, and the test and reference drug products should dissolve rapidly, that means 85% or greater in 30 minutes or less, under mild test conditions in pH 1.2, 4.5 and 6.8, and the test product and the reference product should meet the profile comparison criteria under all test conditions. Then next we see the segments and pH of that of some parts of body. The first is preprandial stomach. The pH is 1.8, that means 1 to 3, and duodenum the pH is 6, that means up to 4 to 7, and third is upper jejunum, pH is 6.5, that means 5.5 to 7, and lower jejunum pH is 6.8, that means 6 to 7.2, and upper ileum the pH is 7.2, that means 6.5 to 7.5, and next the lower ileum pH is 7.5, that means 7 to 8, and the proximal colon pH is 5.5 to 6.5. Then next postprandial stomach pH is 4, that means 3 to 6. Then next duodenum 5, pH 5, that means 4 to 7. Then next upper jejunum pH 5.5, that means 5.5 to 7. Then next lower jejunum pH 6.5, that means 6 to 7.2, and upper ileum pH 7.2, that means 6.5 to 7.5. Then next lower ileum the pH is 7.5, that means 7 to 8, and last proximal colon the pH is 5.5 to 6.5. Then next we see the biological classification systems for immediate release product. A biowaiver is applicable for lower strengths when the highest strength is shown to be bioequivalent to the innovator product and the formulations of the generic products are proportional to the highest strength and meets dissolution profile comparison criteria. Then for modified release product, a biowaiver is applicable for beaded capsules when the lower strength differs only in number of beads of active drugs and the dissolution profile is similar in the recommended dissolution test media and conditions. A biowaiver is applicable for extended release tablet formulations where the lower strengths are compositionally similar to the highest strengths and uses the same release mechanisms and the dissolution profile is similar in pH 1.2, 4.5 and 6.8. The bio-waiver criteria described in BCS guidance are regarded as very conservative. Discussions are underway to consider relaxing some of the requirements for bio-waiver of the drug products. These solutions-based bio-waiver exemplify the role of dissolution in regulating pharmaceutical drug products. Then next we see the dissolution profile comparison. In recent years, more emphasis has been placed on dissolution testing within the pharmaceutical industry and correspondingly by regulatory authorities. Indeed, the comparison of dissolution profiles has extensive application throughout the product development process. Under appropriate test conditions, a dissolution profile can characterize the product more precisely than a single point dissolution test. A dissolution profile comparison can be used to develop in vitro or in vivo correlations which can help to reduce cost, speed up product development and reduce the need to perform costly bioavailability or bioequivalence human volunteer studies. Then establish final dissolution specifications for the pharmaceutical dosage form and also for establish the similarity of pharmaceutical dosage forms for which composition, manufacturing site, scale of manufacture and manufacturing process and or equipment may have changed within defined limits. Next we see the methods used to compare dissolution profile data can be categorized under the general headlines. First, exploratory data analyzing methods are graphical and numerical summaries of the data. Second, mathematical methods that typically use a single number to describe the difference between dissolution profiles. Example, FS, F1 and F2 equations. Resignos indices. Then third is statistical and modeling methods are ANOVA, modeling based methods, mixed effect models, Cho and Case model. Then next, among several methods investigated for dissolution profile comparison, the method described by Moore and Flanner is the simplest and widely applicable. Moore and Flanner describe a model-independent mathematical approach to compare the dissolution profile using two factors, a difference factor F1 and similarity factor, that means F2. Both equations are endorsed by the FDA, Food and Drug Association, as acceptable methods for dissolution profile comparison, but the F2 equation is preferred. The equations are F1 is summation of RT minus T1 upon summation of R1 into 100%, and F2 is 50 log 10 to the in bracket 1 plus 1 upon n summation of Wt in bracket R1 minus T1 raised to minus 0.5 into 100 bracket complete, where n is the number of dissolution time points. RT and T1 are the reference and test dissolution values at time T1, and W1 is an optional weighting factor. The F1 equation is the sum of the absolute values of the vertical distances between the test and reference mean values, that means RT minus TT, at each dissolution time point expressed as percentage of the sum of the mean fractions released from the reference formulations at each time point. The F1 equation is 0 when the mean profiles are identical and increases proportionally as the difference between the mean profiles increases. The F2 equation is a logarithmic transformation of the average of the squared vertical distance between the test and reference mean dissolution values at each dissolution time point multiplied by an appropriate weighing, that means Wt in bracket R T minus T2 bracket square. The transformation is such that the F2 equation takes values less than 100. The value of F2 is 100. When the test and reference mean profiles are identical, value of F1 between 0 and 15 and F2 between 50 and 100 ensures sameness or equivalent of the two dissolution profiles. This range of F2 values equates to an average difference between mean dissolution profile of up to 10%. In addition to this criteria, the FDA scale up and post approval changes modified release guidance states that the average difference at any dissolution time point between test and reference mean profiles should not exceed 15%. The main advantage of the F1 and F2 equations are that they are easy to compute and they provide a single number to describe the comparison of dissolution profile data. For modified release dosage form including extended or delayed release dosage forms, dissolution profile data would be generated at a number of time points, typically 5, until either 80% of the drugs was released or the dissolution profile reached an asymptote. In contrast to modified release dosage forms, dissolution profile data of IR may be generated at fewer time points. The typical USP requirements for IR dosage forms are that 74 of an administered dose based on a mass balance determination or in comparison to an intravenous reference dose, and an IR drug product is considered